So the oxidative phosphorylation comprises of four complex protein complexes, if we can see down here. There's also a quinone pool here that's heavily involved in the transport of the electrons from the beginning of complex 1 to the end of complex 4, and the pumping of protons out of the matrix into the intermembrane space. So as you can see, these are not small proteins. These are rather big proteins with the succinate dehydrogenase being the smallest one. So cytochrome C is not really a classified as a big protein. I would say it's a small one. You could also see here the number of subunits and the prothesis group. So that's what we discussed earlier, that there is these groups that are covalently bound to the protein complex. And there are the FMN domain, the flavone mono NADH oxidase domain, the FAD. There are hemes. There are some ion complexes, and there's different heme complexes and different ion complexes involved in this. An overlook, there's two different figures here that I'm showing here. It's basically showing the same, that you have complex 1 here, which is a large bulky molecule with NADH coming in, being reduced to NAD, and that then the electrons is transported through here, through the quinol loops, the succinate circle of FADH. The complex 3, which have like a very complex transport of electrons. So receive 2 electrons from here and 2 electrons from here, which is where the 2 electrons from the 6 ones here, there's only 4 transported through. 2 of them disappears into this channel here and goes over here and come out here. Now, and then there's the last complex, which is the complex 4, which is where the oxidation of water takes place. No, the reduction of oxygen takes place, sorry. Finally, we have the ATPase, or the ATP synthase, that synthesize ATP from ADP and phosphate. So let's look at it. The electron flow through the systems is this way. You can see the drop of the negative or the energy levels. The free energy again is going down, as we've seen before. So complex 1. What is the size of complex 1? That's a bit different from cell to cell. No, from organism to organism. And so around 900 and 900 plus, as I said. So that's for most eukaryotic cells. It's a oxidoreductase. It oxidizes NADH into NAD, and in the end reduces coenzyme Q into the quinol. So the quinol form, as you see here. Now, it also uses a flavonomononucleotide, FMN here, to transport the electrons inside the molecule, and there's this iron sulfur complex that we'll get a closer look at. So let's look at it a bit. So first we get the NADH here and the FNN here and the iron sulfur complexes, and in the end we get the a quinone. So this is the FMN. So we get a flavin mononucleotide here with the semiquinone intermediate. So the semiquinone is indicated here by this ionization here. So the electron you see here, and then in the end you get a double bond formed here. So with an extra double bond inside this ring system. This indicates that the reduced form here, so in the reduced form we have 2 protons bound to the nitrogen, and in the oxidized form we have these 3, or you could say trivalent nitrogen forms here. So that's the FMN. So this, and where does these electrons come from? They are donated from the NADH that is then oxidized into NAD. So with this reduction here leads to the oxidation of NADH. Traveling through complex 1, the upper part, there's a bound of the there's a binding of the FMN to an iron sulfur complex, and that iron sulfur complex you can see down here. So it indicates here we have like cysteines that donates to sulfur, and we have an iron in the middle. So we have here we have a depiction of 3 different iron sulfur complexes. Again, that is 3 different depending on which organism it is in, but the principles are the same. The electrons are donated from the FMN, so the FMN is oxidized back to this oxidized form and reduces the iron 3 to iron 2 in these complexes. So that's all in complex 1. In this figure, we have 1, 2, 3, and 4 here as complexes. So these are not just complex 1. Now, in the end of complex 1, we have this coenzyme Q reduction, also known as ubiquinone, and the quinol or ubiquinol form of it, as I indicated earlier. So this ubiquinone is then reduced into the ubiquinol. So instead of having 2 keto groups that we see here, we get 2 alcohols group as shown over here on your right-hand side. Again, in between, we have this semiquinone intermediate on the oxygen this time and not on the carbon. And that leads to that we get an overall reaction from NADH plus ubiquinone to NAD plus quinol. And we get release of a proton. Now, this proton can then be transported into the matrix of the mitochondria. At the same time, so then we get this ubiquinone 2- form here that picks up basically 5 protons from the matrix and forms the ubiquinol, and again here transports the protons into the cytosol. Again, so that way we create a membrane potential. So we pump out these protons from the inside of the mitochondria to the outside of mitochondria. So that's what this should indicate down here. So that was the first complex we had here. We got 4 protons here, and we got 2 protons that we just saw bound to the group biquinol, as you see here. The next step is on complex 2. So in complex 2, FADH2 is oxidized into FAD, and another ubiquinol is formed. So another ubiquinol molecule is formed here. So how does this look like in more detail? Well, again, we have inside the succinate dehydrogenase, we have an FAD that is reduced to FADH2, and then during that oxidation, we again have a iron sulfur complex that is reduced while this is oxidized back to FAD. It's reduced and transport the electrons inside here. This leads to a reduction of a ubiquinone to ubiquinol, and as we just saw in the previous description, ubiquinone to ubiquinol requires protons to happen. And that way so this is not a charged ubiquinone, so it's just ubiquinone by itself, so it's only 2 protons that will be picked up to ubiquinol. So in that way, we now have 2 extra protons bound to as a ubiquinol that we can transport over the membrane. So complex 3, so the main part of the complex 3 is a oxidation of the ubiquinol to ubiquinone and a reduction of cytochrome C, the cytochrome C complex. While this is going on, we get a translocation of 4 protons. So the 4 protons are then released into the intermembrane space. So in principle, we would have 4 protons released into the space, but at the same time, we take up 2 protons from the mitochondrial matrix, and so we actually get a release of all 4 into this, but 2 of them are kind of like put back into the ubiquinol complex here. So the ubiquinol cycle is rather complex, but we'll get into that. But so this is the principle behind this. So the electrons are either used for the reduction of cytochrome C or for the reduction of ubiquinol down here, and then that is reduced into ubiquinol. So that's why you don't have a release of 4, but without uptaking 2 here. The structure of this complex, 11 subunits, but you can see there's a couple of heme sites. There's a heme C here, B here, and well, BL here, and BH here. You can also see the heme C over here. The heme sites are important part of the reduction elements. And so the heme C1 here up here is the major reducing part for cytochrome C. And we also have you can hardly see it, but there is actually here a iron sulfur complex. So as I said, the ubiquinol cycle was a bit complex. So it's cut into basically 4 parts, where we have a part that starts with the first one with ubiquinol here, which is oxidized into ubiquinone. So that releases 2 protons up here and 2 electrons. 1 electron is transported further downwards, and 1 electron is transported via the iron sulfur complex into the reduction of cytochrome C. This also, of course, releases ubiquinone as before, and that can be reduced into this semiquinone complex. Now that reduction requires an electron, which is carried through the heme B, L, and BH complexes. So this is the first round. So we have at least 2 ubiquinols coming from the other elements into this complex. The second ubiquinol molecule again comes into a cycle here, and again this is oxidized into ubiquinone. Again, we get a release of 2 protons, but now we have 1 electron again reducing cytochrome C in the iron sulfur complex, and the second electron is then used to further reduce the semi-quinone molecule we had up here. So that one is then further reduced to quinol by the uptake of 2 protons from the matrix. So that way you can say we transport 2 protons through and we release 2 protons from previous uptakes in the systems. Now this double cycle of quinones is also shown over here in 3 different forms, in 3 different reactions, what's actually going on. But in principle, it ends up with that 2 quinol molecules here is reduced to 2 quinone molecules, where one of them is is oxidized into 2 quinone molecules, where one of them is reduced into quinol as part of this process. So that's the Q cycle, also described in many other places, and I have tried to see if I could find a video where it's also been described for other people with an animation, and it should be now on Did You Learn? Part of this is the cytochromes. So the cytochrome structures, maybe you remember that, is a four nitrogen molecules that form a complex with iron. And these are basically the same heme structure, whether it's hemoglobin or cytochrome C or any other cytochrome where you have heme part. So the principle is behind the same. You have these pyridine structures here that are conjugated in a ring structure somehow. So the ring is not always structured like this. It could be different bonds in between, but the nitrogens will always be formed like this. And then you have various side chains. You have hydrophobic tails. You have basically nothing here, and you have protein links and many other parts where this is taking place. But this is the core heme structure, and this is where we have the reduction of iron 3 to iron 2 and oxidation of N from iron 2 to iron 3. So the cytochrome C, which transport electrons here between complex 3 and complex 4, is a very conserved protein. If you look at it, or at least the functionality is very conserved. But you can also see, so we have here different bacteria, and we have a tuner, so actually the fish, cytochromes here, and you can see that we have this alpha helix domain here and this alpha helix domain here. They actually occur in all three of them. We also have the same situation of the heme structure inside cytochrome C is here, you have it here, and you have it here. So as you can see, this is a very conserved structure. So that means that the functionality of transporting electrons from complex 3 to complex 4, which is the main task of cytochrome C, is something that's taken place for many, many years, and is very vital for the understanding of biochemistry, in this part of biochemistry, at least the oxidative phosphorylation. So the final complex is cytochrome C oxidase. So it basically oxidizes cytochrome C from its reduced form back to its oxidized form. Here we can have some heme elements part of it. We have some copper ions that are part of this complex, and several other subunits. So the oxidation is, well, the oxidation is a bit, well, the oxidation is straightforward because it is an oxidation of iron 2 to 3, but the reduction that is accepting this is a copper bridge that is broken and reintroduced in this peroxide bridge. And I will, in the next slide, actually show you how this is going on. So the reaction with complex 4 is that we form a peroxide bridge here that we break, and then we form some water molecules. So the first part is shown as up here, is that we get an oxidation of cytochrome C that releases 2 electrons into the system. Those 2 electrons then reduces this the iron in the heme and the copper, and those need to be oxidized back by a reduction of oxygen. So the oxygen is reduced and form this peroxide bridge. So this is where the binding of the oxygen forms a reduction of the neutralization of the reduced ions here. Now, then we have a peroxide bridge in the system. We get the second cytochrome C. So remember, we had actually had 2 cytochrome Cs from complex 3. So the second cytochrome C comes in, is oxidized, release 2 electrons that go through the system, and they, in order to be reduced, pulls in 2 protons from the matrix and forms 2 alcohol groups. Those are pretty unstable, and in the next step, these will be cleaved off and pull in another 2 protons from the matrix and establish 2 water molecules. So by cleaving the oxygen bond here. And then we are back to where we started, and we can go in another circle again. So in principle, first we form a peroxide bridge, then we cleave the peroxide bridge into two alcohols, and then we cleave all the two alcohols by forming water. So this is a hydroxylation step here. The overall net reaction is that we get cytochrome C, four cytochrome C reaction for one oxygen. We have 8 matrix protons that is then released into water, and the 4 of those protons is released into the cytosol, whereas the other protons are sitting here in the water molecule. So this is the final complex in the electron transport chain, and shows that we have now transported a lot of protons. We created a proton gradient, and we'll come into that in the next video.